Hi everyone, I'm Josemary on behalf of GenScript, and today we are interviewing iGEM Tuebingen. iGEM Tuebingen, can you tell us more about yourselves and your team?
Well, I guess we can start with the fact that as a group of about 20 students, we represent many different fields. For example, ranging from biochemistry and also some plant biology, nanoscience, as well as mathematics and rhetoric and media studies. So it's quite a broad range, not only biological subjects. That's one thing that I really like about our current team. Maybe someone else?
It's actually like pretty interesting thing because we do not only plan to do some stuff in the lab itself as part of the problem in which we are interested in, is that it's also a social problem, and we want to raise awareness via creating an own podcast. And therefore, it really helps that we have some people who do actually study the subjects which come close to this kind of stuff, because as scientists, we don't really have much of experience working on such things. So it's really helpful.
So can you tell us more about the issue your team is tackling and what inspired you to make an impact to tackle the specific issue?
So the main issue we want to tackle is antimicrobial resistance, which can occur due to overusage of antibiotics in medical treatment. And our idea was to use a special class of antimicrobial molecules, which are called AMPs, antimicrobial peptides. And this is the main theme of our current project. Maybe someone else can go a bit into more detail.
I can try. As with any other proteins and peptides, there are many different classes, many different types doing all sorts of interesting stuff. And what caught our attention were the so-called cyclotides, which come from various plant species, where they are thought to be part of the defense mechanism of the plants to various either bacterial disease, but also they have been shown, for example, to play a defense role against insect herbivores, which is kind of cool. And what makes them very interesting is the fact that they create this cyclic backbone. So it's not like a typical protein, which is kind of this linear structure, which then falls into some sort of 3D structure, which then fulfills its function. However, like the N and C terminus actually get connected via these, I think it were like 4 different cysteine bonds. And therefore the structure gets very stable. And so our initial idea was that if you look into different databases, you get like something between 700 and 800 different cyclotides theoretically, which have been described based on some genomic searches, but only very little percentage of those have been already actually characterized by experimentation in the lab. And so our initial idea was, okay, so these have been shown to have these antimicrobial properties, and it would be kind of interesting to test more of those. However, the issue which we faced and afterwards and realized a bit later on was that they also have hemolytic activity, which correlates with the antimicrobial activity. So the more antimicrobial the peptide was, the more hemolytic it was as well, which makes therapeutic use, in which we were interested, rather unpleasant for the person who would take it as a pill potentially. so then we had to think about doing something else, and I think it was Eric who discovered this one paper by Professor Craig from Australia. From what was it? Was it Calgary? No. What was the city again?
Yeah. So the next idea was then to just use another method which a researcher from Australia has had discovered or established, and this is our goal for now. So that we use a scaffold, a cyclotide scaffold, which has regions called loops, which can be exchanged. So there is normally a protein or amino acid sequence, which you can replace completely, and the cyclotide still works and gets produced by an organism. And now the idea is, okay, we have these super stable cyclotides, which are so the scaffold of a cyclotide is not antibacterial, but if we take this stable scaffold and combine it with a antimicrobial peptide, so we graft it into this structure, we have both a non-hemolytic AMP, and which is also, because it's fused into this cyclotide scaffold, which is therefore really stable under many conditions. So we can then apply it in clinical treatment.
And with further development, so you said you would modify a section of the cyclotide scaffold. So with further development of the new cyclotide, what would that look like, for example, for medical applications or mass production to make it accessible for the public? What would further development or industrialization look like for your project?
Now the thing is that if it works as we expect, it would be rather easy cloning system where you would already have this vector in form of a binary plasmid, which can be used in E. coli as well in agrobacteria, where you would have all of the important parts already in there, and only thing which you would need to exchange if you want to test a new AMP, which would potentially show good therapeutic potential. However, wouldn't be very stable, so that you could graft it into this backbone very easily and do all of these characterization steps rather quickly to get to results which would say that the therapeutical use is potentially viable. And if that's the case, I mean, this system which we have could be potentially further adapted for mass production. Not in a typical way as we are used to with different biological products, which are now mostly being manufactured with E. coli and bioreactors. However, you could employ plants for this step, which would be kind of interesting from the ecological perspective as well, because you don't need all these bioreactors, which take a lot of energy, and safety issue would be probably lower as E. coli, even though when modified are S1 safety standards, but like typical E. coli are actually S2, if I recall correctly, and so on. So there would be quite an interesting potential in that sort of way. I mean, the way how we want to test it in the labs to make it very quick is to employ so-called transient expression. So you take the agrobacteria and transfer, infect the tobacco leaves with them, and that way these chimeric peptides or proteins are being expressed there. However, they don't get into the next generation of these plants. However, if you would be interested in creating this in mass production, you could still create a stable line, which this then express in high efficiency, and you could just simply after it has grown, extract it from its grown leaves. So this is like the big idea, but I don't think we will be able to do anything like it. We are only making like a proof of concept. This is something which, if it would work, would be probably done in more, I don't know, 10 years, maybe more.
Yeah, but I think the aspect that we use like in plantar farming is like quite interesting about our project as well. And I think it's like an branch of production of peptides or proteins right now, which is like getting bigger and bigger, and more people are looking into it, even in the industry. And so I think it could be really possible with further development, of course, of our project to use our expression system to produce something like in industry scale.
So you also mentioned that you have a lot of side projects within your bigger project to tackle the issue. So can you tell us more about how you're using human practices to bring awareness to synthetic biology and what's really going into your project and the impact that it could have?
Yeah, so right now I think our biggest human practices project is our podcast, in which we interview experts on different fields. And for example, until now, we interviewed our like primary PI of our project, who is like the head of the nanoscience program of our university, and we also talked to him about the ethics behind synthetic biology, because in the nanoscience program, for example, there is an ethics course included, and so we talked to him about that as well. And that's how we want to go on, ask experts about their opinion, first of all, on our project and what they think about it, and what they have in general for what they think about the ethics behind synthetic biology, and what issues they have to tackle in their normal research with the ethics and everything. And then another thing we are planning is to do a panel discussion. I think that's the right word. And where we also then have like other guests, and we can use what they say or what they discuss about, also to reflect about our own project and see what we could maybe change or have to change to make it more ethical or make it more useful, and to help to raise awareness, for one thing, about antimicrobial resistance and the overuse of antibiotics, and secondly about the ethics in general behind synthetic biology, and not only our project.
Here, of course, we would also add people, for example, from the industry. So our idea is to combine the different points of view, so that we have one representing what does the science says about if it works, then the ethic component, which is very important if you want to also use it in a secure way. That's also a big part of being a scientist, but also a big part is to understand how does the industry work. So they should be also part of our discussions, and possibly also about the podcast. And if possible, we would also try to look at which communities or who are the patients that are normally affected by antimicrobial resistance, and how would they react if we tell them we are creating for you this new treatment. Because it's good if we have a treatment, but the first step is that the patient is also willing to take it. And for that, we need to work in all this field of what is synthetic biology, and we're not trying to harm you, because a lot of people is very scared about it. Often they don't understand what all those special words mean, because if you don't have that background, it's very difficult to find yourself there. So idea also with human practices is to understand what does the public say about our project, and how can we make it fit better to them, and also how can they influence our project, because at one point you start being in a bubble and only thinking about what we want to achieve and not what others would like us to see. So that's also the idea behind human practices and behind those two projects and every other developing in the coming weeks.
And is there anything unique about your approach that you would like our audience to know about your project?
I guess the most interesting point is that we use plants for expressing our proteins. So, I mean, the approach of using antimicrobial peptides it's not like a big revelation in science. But I guess the combination of an antimicrobial peptide and then using these plant-specific cyclotides, so these also stem from plants, and then expressing them again in plants makes it kind of a intuitive, but also I think quite nice idea and concept. And then just using plants which can be scaled up in production as you like, and can be also managed in a secure environment. So as long as these plants can't get into the environment, I think the production can be handled quite well. So I guess this possibility of a new way of producing antibiotics is the thing that strikes me the most. Maybe someone has another idea, but I guess that's, for me at least, the most special part about our project.
I mean, if it works the way we hope it would, it be actually pretty interesting, because most of I mean, antimicrobial peptides as a potential treatment, this idea has been around for quite a while. But as already mentioned, like most times when you try to create some peptides or proteins in big scale, you employ bacteria. However, as you want to create a peptide which has antimicrobial properties, to create it in bacteria is rather problematic. And also, many of these peptides which have been so far described don't necessarily stem from bacteria, which are prokaryotic organisms. However, they come from eukaryotes, and the transcriptional machinery is therefore a bit different. You have a lot of post-transcriptional modifications, which are part of these proteins and are required for their correct function, which is hard to mimic in organisms such as E. coli. And therefore, if you bypass all of this by using an eukaryotic organism such as tobacco, which we plan to use, you bypass all of these problems and potentially have a new solution. And yeah, I guess we have to see if it works or it doesn't. But, yeah, I'm looking forward to it.
Well, iGEM Tuebingen, thank you so much for meeting with me today to tell us more about your project. We wish you the best of luck in the iGEM 2021 Jamboree and in our GenScript iGEM 2021 sponsorship competition. Once again, thank you so much for meeting with me, and best of luck in the competition.